Hi, good afternoon. So we'll get started. So already I've been told by one August unnamed member of the audience today that this is the Golden Gate Bridge. But you all know that it isn't. It's the logo of this course. That's the Brooklyn Bridge. And today's subject is particularly well suited because the thesis of a bridge, of course, is that things are different on both sides. And when you build the bridge, they become different on both sides again because they become mixed. And so that's the dilemma that will be discussed today. The RAS oncogene, the most commonly mutated oncogene in all human cancer, with an enormous amount being known about its structure, mechanisms, and so forth. And then on the other side of the bridge is it's mutated in so many different human cancers, and the problem is how to put it together, particularly in some therapeutic manner. So where is... Oh, I have to do this. So just by way of brief introduction for those who are not RAS aficionados, RAS is originally named for two rat sarcoma viruses, which were identified by two different investigators, Harvey and Kirsten. And then the genes responsible for these viruses, for their transforming properties, were discovered here at NIH, mainly by Ed Skolnick and his associates. And then later they were found by many, many others in various cancer cells. And they were transforming when put into tissues and cells. And then in 1982, several people, including, if I recall correctly, I think Mike Wigler at Cold Spring Harbor, was studying a neuroblastoma and found the third member of the family, which is called NRAS because of that. The three human RAS genes code for very similar proteins, about 188 to 189 amino acids. And significantly, they are GTPases, which, as we will hear, control many aspects of intracellular signaling networks, and when overexpressed, are associated with growth, differentiation, and transformation. Now, it's interesting, and this is the challenge. The frequency of hyperactivated mutations of each of these RAS genes occurs in different distributions with different cancers. In some, like pancreatic cancer, Kirsten RAS is overactive, is overexpressed, is mutated in almost 100% of the patients, and others. We'll hear more about this. So the big question is, given all this information, about which we'll hear much more, why is there this big diversity in RAS genes? How does it happen? What is their potential? What's their differential function normally and in malignancy? And given this propensity to be overexpressed in different malignant diseases, are they really therapeutic targets? And if so, where do we stand? So the answer to that is to build a bridge. And the bridge that's being built here is the RAS Central at Frederick, which was started, I think, a year and a half ago. And Frank McCormick, who's the director of it, is one of our speakers today. This is an effort about which we'll hear more, but which serves to try and answer bring together information not only from NIH but all over into this important question. So we're very fortunate to have two experts discussing both sides of this proverbial bridge. So this first speaker is going to be Susan Bates, who I suspect is known to most of you. Susan's the head of the molecular therapeutics groups in the developmental therapeutic branch of the NCI. She graduated from Arkansas School of Medicine, trained in Georgetown and in oncology here, and she became tenured in 1992. She's very well known for her exciting work on drug resistance in malignant disease. And Susan is going to discuss the sort of status of the RAS genes with respect to human cancer. And then Frank McCormick will take up the theme. And for those of you who don't know, Frank is emeritus professor at the moment of medicine from UC San Francisco and from the Comprehensive Cancer Center there. He received his PhD degree in Cambridge and then was a fellow at Stony Brook on Long Island with Seymour Cohn, and then at Imperial Cancer in London with Alan Smith. And then when he went to California, began his career with three leading biotechnology companies, almost from their beginning, Cetus and then Chiron, and then he formed Onyx company. Now, the goal of all of this was to study the malignant process and to develop therapeutic agents, some of which he did develop, particularly sorafenib, which has been approved for, I believe, renal cell cancer and I think hepatocellular cancer. Frank's a member of the National Academy and has received untold honors, which I will not go through to relate to you, except to say that he's been a leader in this field and very productively, and we're very grateful that he is here. In his present position, he's oscillating between San Francisco and Frederick because he's the director of the RAS initiative, which I briefly mentioned before. Susan.
All right. Thank you. Well, I want to say thank you to Wynn for inviting me to do this. When he invited me, I said, well, I'm not really an expert in pancreatic cancer or lung cancer, but I said, I'm a medical oncologist, and medical oncologists have basically been fighting RAS for, the entire time we've been medical oncologists. So I framed this to be basically a lead-in. I told Wynn I'm the warm-up band for Dr. McCormick's talk because he's working with the RAS initiative to come up with therapies for RAS cancers. So what you're to think about here is, what are we going to expect from that initiative? What can we hope for? What can we expect? So the first question, is it the Holy Grail? I told Tito, it's the Holy Grail. He said, Tito's, always questioning things, said, it's not the Holy Grail. So there we had a complete opposite opinion about RAS. And you can decide at the end, is it the Holy Grail? Is it not the Holy Grail? So we're going to cover these things. How often do we find it, and what diseases? What does it teach us for where we find it? What is its role actually in outcome, in whether patients do well or don't do well? What is its role in affecting other therapies? What is its role in the origin of cancer? What approaches have been attempted? And what benefit can we expect from targeting RAS? So we're going to scratch the surface for this. These are all very deep questions about which many, many papers have been written. So this is a timeline that when basically got started. And so one point is, though, that this was 50 years ago. So we've known about the transforming potential of these sarcoma viruses for 50 years, which is remarkable. And just to follow through a few details that I wanted to point out. So very early on, we knew the viruses, we knew they were transforming, we knew that, then found Dr. Varmus actually discovered that a normal mammalian counterpart would be a normal cellular counterpart to the viral oncogenes, that if they're small proteins, that they were GDP and GTP binding proteins, and then that they were overexpression would transform NIH3T3 cells, and then the mutations were discovered, and that just opened an enormous round of investigation. Mutations that were themselves transforming. So you could either make a cell become a cancer cell either by putting in a lot of a normal RAS, or by making a mutation in RAS. But then there were papers appearing that said you can't actually transform a primary cell. Take a really normal differentiated cell. You can't transform it without some cooperating genes. And then eventually the fact that RAS had to be farnesylated to hook into the membrane to actually signal meant that the discovery of inhibitors was on, and that's when medical oncologists really got involved. So that's why I say this is the timeline to the start of medical oncology for RAS. Now, it is a small GTPase, and I'm going to leave the biology to Dr. McCormick, but the points being that it's a very well understood protein, highly conserved. The H, K, and N RAS is highly conserved, but for this 1, 5' region. And then the switch that occurs between RAS off and RAS on is what is actually impaired, so that you can't turn it off once you have the mutations that have been discovered in the viruses and in the cancers. And that's what we'll talk more about. So the incidence in cancer, as Wynn was saying, one of, if not our, I'm not sure it's our highest mutated cancer, but that may be true. It's mutated in 22% of cancers, KRAS is. Some of the others are less. The HRAS is only in 3% of cancers, and NRAS in 16% of cancers. And there's a differential distribution across them in which we do see, for example, in hematologic malignancies, more NRAS and less KRAS, whereas in the solid tumors, we see mostly KRAS. And then HRAS is actually much less common, even though that was the first discovered. So here is the mutations, and the point to be shown here in pink are the cancer-causing mutations, and in green are mutations that we're not going to get into today. Maybe Dr. McCormick will, but these are in what are being called rasopathies, so in diseases that are not cancer-related. And so these mutations are occurring more across the protein, whereas there are 3 hotspots. 97% of RAS mutations are in these 3 hotspots at codon 12, 13, and 61. And it's very rare at other residues and other locations. And I would just also, if anyone wants to do more reading, this is an outstanding review article on RAS by Adrian Cox and Channing Durr. So it's interesting to look, the different RAS proteins have mutations that are somewhat differentially. So for example, the KRAS isoform is mostly mutated around codon 12. That's the most common. Whereas the HRAS is more in codon 12 and codon 61. And the other interesting thing about this is that KRAS is mostly codon 12 mutations found across cancer, but that's not completely, it's not true in every single cancer. But in pancreas cancer, for example, it's almost totally codon 12. And in lung cancer, almost totally codon 12. That turns out to be related to cigarette smoking, and the others are actually occurring in patients who are non-smokers. So one of the interesting things about RAS I mentioned a moment ago is that it's farnesylated. It has to have, by farnesyltransferase, it has to have some lipid groups attached to it so that it can plug in to the cell membrane. And that led ultimately to the first therapy that was actually tested for RAS. And we'll come back to that in a bit. And RAS then signals through at least 6 validated intracellular pathways and signals, at least as far as we know, currently for multiple important things in cancer cells: survival, progression, migration, endocytosis, the cytoskeleton, cell cycle progression, calcium signaling. So really every cellular function is in some way affected by RAS being turned on and left turned on. So we'll focus then on the mutations that are present that have been described in acute leukemia, in colorectal cancer, 33%. Acute leukemia is only 5% for KRAS, 15% so for NRAS. Lung cancer, which is in about a quarter of lung cancers, that number's a little bit low, and then pancreatic cancer, 60%. So what can we learn from clinical data in these specific cancer types that will help us decide what benefit might accrue from a successful RAS initiative in Frederick? That's the question that we have here in front of us today. So this is a nice study of lung cancer, 670 patients that have the KRAS mutation in lung cancer. And it's interesting to note that those who were smokers currently or former smokers have this nucleotide transversion of G to T at that 12 codon. And then that results in typically in the glycine to cysteine mutation at codon 12, whereas the never smokers actually go to aspartic acid. So it's pretty interesting that tobacco smoke causes a typical mutation and a typical outcome. So I think the biology of the differences between those mutations still remains to be really worked out. So one question is, we always thought for years and years that RAS made people have much more aggressive cancers, die much sooner. But there's really not much evidence for that. When we've talked about it in leukemia and colon cancer, lung cancer, we're not really seeing that. When it's put to a test where you look at a specific cancer type, it seems that a specific subset, it seems that there's no real impact. So this is a Kaplan-Meier curve for disease-free survival and overall survival. And this is the way we typically look at the outcome in cancer patients. And here you can see blue for the wild type and gold for those who have mutations. Those curves are overlapping. So no impact on outcome in lung cancer. And then so the question is, could it be a therapeutic target in these quarter of patients with lung cancer that have a KRAS mutation? Now, lung cancer in recent years has become incredibly interesting because it's been subsetted into different mutations that are called oncogenic drivers. So we have EGF receptor, epidermal growth factor receptor, which is driving a proliferative pathway, ALK, and many, many other small percentages, but nonetheless, where we found, for example, ALK is 5%, you add an ALK inhibitor, patients get marked benefit from that. So adding an ALK inhibitor gives marked benefit. Adding an EGF receptor in the EGF receptor mutant lung cancer gives marked benefit. It suggests that we should get marked benefit when we inhibit RAS in lung cancer. But we don't know. That's what the question is going to be. Now, the epidermal growth factor pathway is activated in lung cancer, as I just showed you, at multiple levels, EGF receptor, RAS, BRAF. So it's actually been tried already, a MEK inhibitor, which is blocking this signaling pathway. Now, if we have KRAS mutated, I showed you there are 6 validated signaling pathways, but this one, anyway, is one of the most important because it leads to cell proliferation, or we thought it was important. So what does MEK inhibition do? Well, that's selumetinib added to chemotherapy, and this is a waterfall plot, which is how we show whether patients' tumors shrank or didn't shrink. So here are tumors increasing in size, here are tumors decreasing in size, and what you see is that selumetinib gave a few responses, but really not much. This is not a home run. This is nothing like ALK inhibitor or even the EGF receptor inhibitor. But it is MEK, it's not RAS. So we still can hope that we get major benefit from RAS inhibition in lung cancer. But this was a little disturbing that we didn't see more from MEK inhibition here. Now, if we move on to colorectal cancer, one thing that's interesting here is that colorectal cancer arises from, at least familial and many of the syndromes, arises from early DNA defects.
I don't know if this is the correct one or not.
The Anthony is, that's the correct one. She said the same one.
And so,
that's why I didn't send it to
RAS doesn't appear as a mutated
I'd say we're good because we're actually
later
OD
on.
building 551.
We pause. Put the recording on.
Okay.
Whoops. So it doesn't appear until a little later on. And then other mutations accumulate to get the final very invasive cancer. So how important are mutations that arise later? Can we say which one is truly oncogenic? It's an accumulation of mutations for sure, but what does it mean that RAS arises about halfway through that process? And another point that says RAS may or may not be the most critical thing in colorectal cancer, although it's prevalent in 30%, is that when people have looked at mutations in lymph nodes, distant metastases, and the primary tumor, it's not 100% concordant. So you don't always start out with a wild type and end up with a wild type, although generally you do. And you don't always go from mutant to mutant, although generally you do. But there are examples where you have KRAS appearing later in a new clone at the time of relapse. And that's a very common finding, actually, in leukemia, which we'll get to in a few minutes. So then what about clinical trials, and what can that tell us about how important KRAS is in colorectal cancer? So there's been 2 drugs used to inhibit the EGF receptor pathway, cetuximab and panitumumab. Are we okay?
Yep.
Okay. All right. So cetuximab was shown to benefit colorectal cancer, and the FDA approved it in 2004. They approved it with not very many months benefit in terms of disease shrinkage or disease control. But nonetheless, they approved it. It wasn't selected for particular patient subsets. But then, 4 years later, a group discovered that in the trial that was done on which the FDA approved cetuximab, they found that KRAS mutations actually do not give benefit to patients who are receiving cetuximab. So here is what happens: absolutely no benefit at all from cetuximab, which is inhibiting that MAP kinase signaling pathway. No benefit in the presence of the KRAS mutation. And if you have a wild-type KRAS, yes, you do get benefit from adding this EGF receptor inhibitor, cetuximab. So that was one of the first clues that we have to be careful with inhibiting the EGF receptor pathway, because downstream of that, we're going to have alterations that may affect whether you respond or not. And so here you have EGF receptor signaling through KRAS, BRAF, MEK, and ERK, which I showed you as one of the proliferation pathways. And you can imagine that when you try to inhibit above that, the EGF receptor, then you're going to not be successful if you've got your signaling starting below at the level of KRAS. And then that's actually also true, which I'll show you, if you have mutation in BRAF. So if any mutation below the level of that receptor is going to be problematic for treating with EGF receptor. So it's interesting, I think, and this is a little aside, but the ASCO then issued a provisional statement saying maybe you should think about testing for KRAS. That was in 2009. I don't know why they were so equivocal about it. And then it took another 4 years from that first publication to actually end up with an FDA-approved medical test, or they call it a medical device, but a testing for KRAS mutation. And this actually is now done and recommended to be used in combination in colorectal cancer, in combination with either cetuximab or with panitumumab, which is another EGF receptor inhibitor. And another interesting point is that this KRAS test that's FDA approved doesn't test for every mutation. It tests for multiple codon 12, which is appropriate for many cancers, but not for all cancers. So you can't think, I'm going to always use my KRAS test, because you will miss the codon 61 and you would miss other mutations than this group right here. But it does pick up the one that we talked about being in lung cancer. And so this has been reproven several times, even clinical trials combining multi-agent chemotherapy called FOLFIRI with KRAS testing found only patients benefited who had wild-type tumors. So it's very clear, you have mutation, you should not be treated with anything that inhibits the pathways above KRAS. So, and this, it's not beneficial, and it could even do harm. So I think that this fundamental fact that you have this downstream aberration keeping the cetuximab from working implies that the RAS pathway is critical in some way in proliferation in colorectal cancer. Here's the BRAF result where you have treating with cetuximab. If you have a BRAF mutation, also nothing, no benefit. So then they recently have looked for other types of mutations in the KRAS wild type group, trying to see could we have had a better response rate if we got rid of all those downstream mutations. And they found indeed within that wild type group are a number of other mutations. BRAF, mutually exclusive with KRAS, and NRAS, exclusive. So you don't have the two RAS mutations in the same patient. And so then what they looked at was the response to cetuximab in these patients who had progressively removing each of those mutations, so removing the patient population. So you start out with a 24% response rate, not great. Then look at only wild type KRAS and 36% response rate. And then remove the BRAF mutated disease, remove those with NRAS mutation, and remove PIK3CA mutation. And then you end up, once you removed all the mutations, 50% response rate. So you can double the response rate if you basically define your mutation spectrum in your cancer. So this is where the whole idea of personalized medicine really starts to be, this is true. You should not get cetuximab if you have any of these mutations. Then moving on to pancreatic cancer, this is different from colorectal cancer, different from lung cancer, in that when people have really tried to look very early, when it's just the dysplastic epithelium, when it starts to be what they call an in situ PAN1 in the very earliest point, you already see KRAS mutation. So this is either true and the first mutation, or something hasn't been found that needs to be found. But I think that this is accepted, that in pancreatic cancer, the first mutation is the KRAS mutation. And in fact, at early pancreatic intraepithelial neoplastic lesions, 95% have KRAS mutations. 95%, the vast majority. And when you look at progressively worsening intraductal lesions, PAN1N, A, B, 2, and 3, there's not much of an increase, which all of that's consistent with the idea that KRAS is fundamentally the important oncogenic driver and the source of pancreatic cancer. So that suggests that it is fundamental to cancer oncogenesis, pancreatic cancer oncogenesis, and this is really the proving ground. So if the RASH Initiative is going to come up with a drug, it better work in pancreatic cancer. That's like a huge challenge to you because this is one of the most drug-resistant cancers we have. So then AML, though, brings us a really interesting and different story. So AML, this is a variety of patients. You don't have all these mutations in every patient, obviously. But they found a number of different mutations and then have classified these mutations in acute leukemia. So mutations that affect DNA methylation, that affect chromatin remodeling, chromatin modifiers, transcription factors, across the board mutations. And they've also found mutations in RAS, as I've alluded to, which I'm not seeing here where others. And also mutations in KRAS, more rare, but also KIT, which is a kinase, and FLT3, which is a kinase. So these are kinases that have activated signaling versus these are epigenetic mutations that people like to say affect the landscape. So it's interesting when they've done this kind of Circos diagram that the signaling mutations are linked. You never have two signaling mutations. They're non-overlapping, but they are linked to these more epigenetic, these landscape mutations are what people are calling them. So you need an aberrant landscape in cooperation with the signaling pathway mutation. So every time you have a signaling pathway mutation in leukemia, you have a landscape mutation. And so what a number of people, Tim Lay and a number of other people have done is start to say, these landscape mutations are actually occurring first. So a couple of lines of evidence for that. One, in elderly patients who do not have AML, they find these landscape mutations, and they find clonal populations. So almost suggesting that if patients live long enough, they would get leukemia rising in those clonal populations that have these landscape mutations. And here's an example of a patient with leukemia. So they're calling this landscape mutation set the founding set. And the signaling, the proliferative signal is coming from the FLT3 mutation. Well, this could be NRAS or KRAS here, because the subclones are the ones that then are going to outgrow and the patient's going to have frank leukemia from. And when you treat the patient, and then after 30 days, patients are in remission, the proliferative genes that are mutated disappear. They're gone. And this is a number of patients, but the proliferative genes disappear, but those landscape epigenetic modifiers, those persist. So those pieces of evidence, and then here's a different paper showing basically the same thing. When you have pre-leukemic clones, you have IDH1, 2, DNMT3A. These are epigenetic mutations. This is something that affects ribosome biogenesis and chromosome and histone, a histone chaperone. These are there. These are the landscape mutations. Whoops. These are the signaling, and those are just never found in those early pre-leukemic clones. So the current thinking in leukemia is that you have these founder landscape mutation, and then that expands, and then you get a proliferative mutation, and then that expands, and then you have frank acute leukemia. So that suggests, differently from what I said in pancreatic cancer, that RAS is not really the precursor lesion. You could get a great benefit, but you're probably not going to likely cure patients unless you can address the landscaping mutations. And then there's another point that makes it more difficult, and that is that there's a whole superfamily of genes that also are small GTPases. And I would like someone to tell me that they're not involved in cancer, because I don't think that's been proven, yes or no, yet. And so quite a number of them, RAS, RO, RAB, ARF, each of these has a family. Here, for example, the RAS family and the RO family. So we don't know to what extent they're involved in cancer. But they're GTPases and could serve like RAS. And in fact, very recently was reported evidence of RO mutations in T-cell lymphoma, where the epigenetic lesions were already known and people were thinking this is an epigenetic disease, but then suddenly there's a RO mutation. And so the question will be, can we go back and figure out which came first, the founder or the proliferator? So what about therapeutic target? So as we mentioned earlier, RAS mediates signaling through 6 different intracellular pathways. It's well-known AKT pathway, multiple members of that pathway, the MAP kinase pathway, which is the one that we've really studied the longest, BRAF, MEK, MAP kinase. So in a way, you could say we've done some work around this already by using rapamycin, which is an mTOR inhibitor downstream, and we've used MEK inhibitors, and we've not seen home runs in cancer. I mean, they've been used. That's not to say that they've been used in well-defined settings, but they've been used. So that's concerning for how well we're going to do. But one has to hope that when we inhibit RAS at the top of all this, we'll be inhibiting all the pathways, and then we'll have more success. So what have we done clinically? Well, we had a farnesyltransferase inhibitor. So I mentioned earlier that you have to have farnesylation in order to hook RAS into the membrane and then allow that signaling pathway that I just showed you. KRAS actually has a separate, and NRAS a separate prenylation pathway in that it is geranylated. And so that is another way it can get into the membrane. But farnesyltransferase inhibitors were developed and taken to the clinic and studied pretty intensely, particularly tipifarnib and lonafarnib. Now, both of these made it into phase 3 testing in the clinic. And it's remarkable in the oncology literature because there's paper after paper after paper and then stops. It just comes to a big halt. Now, why did that happen? So what they found early in AML was that there were responses in 10 of 34 patients. None of those patients had RAS mutations, but they said, well, maybe this drug works anyway. We don't really care if it hits the target or some other target. We're going to go ahead with it. And but it did show inhibition of farnesylation, and they did find, they used surrogate substrates and were able to confirm inhibition of farnesylation. So the big phase 3 trial that randomized tipifarnib to best supportive care, which could include a chemotherapeutic that's used intermittently in acute leukemia, overall survival is almost identical. And response rate, 8%. So for all that drug development, complete response rate, 8%. And in leukemia, if you don't get a complete response, it's not a good situation. And so 11% overall. So they packaged this up and went to the FDA, to the Oncology Drugs Advisory Committee, who said not enough activity to be worth FDA approval. And so the company continued development for a little while. So this was 2005. And people said again, well, maybe we can successfully do this because there are targets we don't know about. And we're inhibiting farnesylation of a lot of other targets. And we'll keep testing it. But ultimately, someone came up with a 2-gene signature that they said, this reflects sensitivity to tipifarnib or to inhibition of the farnesylation. And so then they took that 2-gene signature, and then they took tipifarnib, which is Zarnestra, and they took that to a clinical trial, selecting patients based on that 2-gene signature, the CR rate, 11%. So it went up by 3 over the unselected patient population. Notice they still didn't pick HRAS or NRAS. They picked this 2-gene signature. So the company decided to terminate further development, but the light at the end of the tunnel, now we're learning so much more, is that they actually have licensed it out for development in hepatitis delta virus, which Wen says, oh yes, I know all about this. So it turns out that the other advanced stage farnesyltransferase inhibitor, lonafarnib, also has a new life in inhibiting hepatitis delta virus. So it's a well-characterized, late-stage, orally active inhibitor of farnesyltransferase, and the enzyme is required for hepatitis delta virus infection, and it uses that cellular process in order to complete its life cycle. And so when patients have received lonafarnib, you have a marked decrease in hepatitis delta virus. So oncology gifts this drug to hepatology. But what we could see happening is if we come back and we say, we understand this process a little better, we could end up repurposing these drugs. But that's sort of a dream. Ooh, you can see we're almost done. So then what about that gold standard? I said pancreatic cancer, we ought to be able to inhibit RAS and if we're going to do anything in, we're going to be able to target RAS, it's got to be in pancreatic cancer. So the farnesyltransferase inhibitor tipifarnib, no benefit. In combination with a chemotherapeutic that's approved for pancreatic cancer, gemcitabine. No benefit in terms of survival, in terms of duration of benefit, or in response rate. So no wonder the company decided to stop development. So the bottom line, it's not clear that RAS mutation confers a worse outcome. I covered that. And the RAS signaling, though, does interfere with EGF receptor blockade, so then it would confer a worse outcome. If you're being treated with an EGFR inhibitor and then you have a RAS mutation, that's not good. But beyond that, its role in oncogenesis is still being worked out. We have the early data that it was not able by itself to transform primary cells, and we have the new AML data that it's not going to transform in the absence of a landscape mutation. And then KRAS appears to be the first event, though, in pancreatic cancer, explaining that difference. KRAS mutant and non-mutant subclones do often coexist. I showed you some evidence for that in colorectal cancer, but definitely in AML. So the bottom line is an important and a critical target for cancer therapy, but its inhibition may not be sufficient in many cancer types. So we need definitive KRAS blockade to answer many of our questions about the role of RAS in the origin and maintenance of cancer. We need definitive KRAS. So you have your mission set out. The KRAS initiative. And all of NIH, really. NIH has been very involved in RAS development all along. And I just want to acknowledge I've had a lot of great collaborators at the NIH and just helping me understand biology and of cancer. So they're all listed here. Thank you.
Susan, that was a great summary of a very difficult, complex field. Maybe we have a few questions. If someone has a burning question they would like to ask right now, otherwise we'll have more questions at the end after Frank. Yes, wait a minute. We need a microphone for you.
I've got.
No, you. Oh, he took mine. What? Thank you.
Hello, thank you. So you showed that RAS is an initiating event in pancreatic cancer, but appears to be accumulated in other types of cancer. What is it about the pancreas that selects for a RAS mutation right off the bat?
I don't know. One idea that's batted around in the literature is that it's due to the bile duct acid being in that area. Right. Well, I mean, right. Well, it does. In the you have gallbladder cancer and you have biliary cancer, and so they are. They do have RAS mutations, but that's not an explanation. That's just one thing that I read. But very good question. Why would pancreatic cancer be so susceptible to this mutation? We don't know. It's not exposed to cigarette smoke, so far as we know. That's another whole area of interest that I think is very important, inflammation. I remember still the day the paper came out with the Rouse sarcoma virus way back when, injected into a chicken, and it didn't get cancer unless you clipped its wing and it got a big inflammation and then it got cancer. I think in human cancer, that's been clear for a long time.
Can I ask, when any of these tumors as lines or as primaries have been put into animals, do they respond to the farnesylation inhibitors or to any of the other?
For many of these, the animal data, the animal models, which we usually use what we call a xenograft model where it's put under the skin, the response is almost always much better than we get in actual patients, much better. So that's also a work in progress, developing better models.
Michael?
Susan, you showed us that in lung cancer, if patients have RAS mutations and you're targeting EGF, you don't get a good response.
And colorectal, both cancers.
And colorectal. If you do the same and get a good response, when the patients relapse with resistant disease, do they have RAS mutations in the relapsed tumors?
That's been reported. So Vogelstein has reported that he can detect RAS in DNA after rituximab treatment in colorectal cancer. Some people dispute those data, but within 3 feet of you, actually.
Susan, what is the thinking about frequent coexistence of mutation and chromatin remodelers and RAS?
What's the thinking about it? I mean, I have no idea then how the RAS mutation occurs, but perhaps this is a hot spot. Perhaps there's something in the chromatin that this is a place that is not easily, that is easily accessed, so that you end up with a mutation. And then once you have this abnormal chromatin, it's even more easily accessed and damaged. So I don't have a sophisticated answer for that at all. But the leukemia data is very striking, I think.
Does inflammation upregulate or alter RAS expression, and does RAS expression alter inflammation?
Yeah.
Do you have the answer for that? Right. That is a good question.
Okay, I think, Frank, if you'll continue, and we'll have time for questions at the end of Dr. McCormick. That was great. That was a wonderful series. I tried to read some of this, and my God, it's confusing.
that's a great introduction. Thank you very much. And thank you for inviting me to come down and talk. Okay, thank you. So I'm going to continue the same theme, but focus more on the molecular biology and the biochemistry of the RAS protein itself, because really we feel that to develop therapies that target RAS directly, we need to understand the function of the protein in exquisite detail. And as I'll try and point out, the mutant RAS proteins that play a major role in human cancer each have unique and distinct properties, which offer opportunities for therapeutic intervention. That'll be one of the themes of my presentation. Let's see. Going here. So this is the lab at Frederick, which is the RAS central hub, as you described it. Has anybody in this audience been to this facility yet? If not, nobody at all? If not, I suggest you visit. It's a very beautiful facility, and there are some 50 people up there right now working on the RAS program to try and find new ways of targeting RAS. This is my lab at UCSF, where I spend the other half of my life. As we've already heard, RAS plays a major role in human cancer, and it's really KRAS that is the major player. This is for reasons we don't really understand, because as we heard from Susan, KRAS, NRAS, and HRAS are very similar proteins, and they're actually expressed pretty much in all tissues. So tumors driven by KRAS also express NRAS and HRAS, and so on. So it's not as if individual tissue types express selective types of RAS. They're expressed everywhere, and yet there's a very strong bias towards different tumor types in terms of which RAS isoform gets mutated. We don't understand that really at all. But KRAS is the major player in human cancer. HRAS, as we heard, is a minor player, playing a small role in terms of numbers of bladder cancer and thyroid cancer. And we also heard from Susan that HRAS actually is a target for farnesyltransferase drugs, whereas NRAS and KRAS are not, because they have a backup system. So there are now renewed efforts to target HRAS-driven bladder cancers and thyroid cancers by pre-selection of patients with HRAS mutations, followed by treatment with farnesyltransferase inhibitors. And we're very interested to find out the results of those clinical studies, because they will obviously could provide benefit to these patients, but could also teach us about the effects of targeting KRAS by analogy. This is the RAS protein, which has been described as the beating heart of signal transduction, because the protein is heart-shaped, and also because it changes between two configurations, the off state, which is bound to GDP, and the on state, which is bound to GTP. So RAS proteins are essentially binary switches, which just turn on and off according to whether they're bound to GDP or GTP. And as I'll show you, one particular part of the protein changes in conformation when it's in the GTP-bound state, and that's where all the downstream effectors jump on and bind. So we have a pretty good understanding of some of the molecular details of RAS and its interaction with some of its partners. This shows a diagram of how this binary switch actually works. So again, on the left side, this is another way of looking at the RAS structure, which binds very tightly to GDP or GTP, and this binding determines whether it's on or off. When these structures were first solved and the biochemistry was first realized, it was realized it was a GTP binding protein, people suggested that maybe we could target mutant RAS proteins using analogs of GTP that would block the mutant protein selectively. But it turns out that RAS proteins bind to nucleotide with extremely high affinity, picomolar binding constants, very slow off rates. And on top of that, cells have a very high concentration of guanine nucleotides, GTP and GDP. So the idea of finding a drug that will compete with the endogenous pools of nucleotide and get into the RAS active site, competing with GTP, was abandoned many, many years ago, although some people are rethinking it with some clever nuances. But that, unfortunately, doesn't seem to be a tractable place to attack the protein. Even though there's a nice pocket there, it's already full of nucleotide, and doesn't seem to be an easy way to get an analog into that site. But anyway, again, the activity of the protein depends on whether you have GTP or GDP. And this is how the switch actually works, shown on the right. So here's GDP with a gamma phosphate to make it into GTP. The gamma phosphate of GTP actually holds these two parts of the RAS protein in a rigid configuration, switch 1 and switch 2, which are these two parts over here. So this is where RAF and PI3 kinase and RAL-GDS, the three effectors that are highlighted previously, bind to RAS in this rigid structure. But if you hydrolyze GTP and this gamma phosphate comes off, this kind of spring mechanism opens up, and both switch 1 and switch 2 become disorganized. And now these proteins don't bind. RAF and PI3 kinase and RAL-GDS don't bind. So GTP holds the whole thing together in a state to which other effectors bind. And you cut the bond, and then it sort of unravels, and then no longer can interact with downstream targets. So that's how the molecular switch actually functions to engage downstream proteins. But things are very complicated, as I'm sure you've already gathered. Downstream, there are a number of effector pathways which RAS in the GTP state can interact with. And even the conversion of RAS from the off state to the on state is a complicated, very highly controlled process. It doesn't occur sporadically through losing GDP or binding GTP. The exchange of GDP for GTP is controlled by large and complicated proteins that only kick RAS into the active state in response to upstream signals. And EGF or receptor tyrosine kinases are the best known signal input into the system through SOS, kicking RAS into the GTP-bound state. But actually, there are many other signals that can feed into RAS through other exchange factors to activate it in cells. So this is a highly controlled process that only happens when signals come into the cell. The off state is equally highly regulated by another complicated family of proteins called GAPs that stimulate hydrolysis of RAS-GTP back to RAS-GDP. And there are a number of GAPs in the cell. The most important, it seems, in human cancer is the NF1 protein, neurofibromin. And we know that to be true because the NF1 gene is frequently mutated in human cancer. In those cancers, the off switch is defective, so RAS can accumulate in the GTP-bound state. Another way of getting RAS into the active state by breaking the off mechanism. So this whole process is extremely highly regulated because this is the fundamental switch which determines whether cells will divide or not, and many other fundamental biological properties. But oncogenic mutations in RAS at codons 12 and 13 and 61 and some others basically lock the RAS protein in the active state by making them resistant to the off-switch proteins, the GAPs. So these GAPs can then, they can actually bind to the mutant protein, but the off-switch is broken. And I'll show you how that actually gets broken in just a moment, because one thought is to try and fix the broken off-switch and somehow trick these GAP proteins into turning RAS GTP into the off state. Sounds crazy, but that would be the most elegant way of fixing the problem. So when RAS proteins can't be turned off by GAP, they get stuck in the on state and therefore become less dependent on upstream signals. They're not totally independent, as I've indicated here, but they're way less dependent on upstream signals and therefore persistently signal downstream. That's why, obviously, inhibiting EGF receptor doesn't do much good in a cell that's dependent on downstream signaling, as Susan indicated. So the problem is then that the switch is stuck in the on state, signaling persistently, and we have to figure out a way of fixing that, either by hitting the switch directly and fixing it, or understanding how to turn off downstream signaling. So just a little bit on the actual mechanism of the off switch. So the structure of RAS and the catalytic domain of GAP from the neurofibromin protein has been solved by Klaus Schefzek and colleagues. And it shows us how the whole switch normally works in cells in which RAS is wild type. And it's a very cool mechanism, I must say. GAP proteins bind to RAS, and they create an active site for GTP hydrolysis. RAS proteins on their own are extremely poor GTPases. But when GAP binds to RAS, it actually creates an active site. And it does so by poking into the active site of RAS an arginine finger, as it's called. So this shows the arginine finger from GAP poking into the active site of RAS. You see coordination here of the glutamine 61 residue, which is frequently mutated in human cancer. And here's the gamma phosphate of water, of GTP and water molecules here. So the arginine finger actually reorganizes the RAS protein to make it into an enzyme so that it can efficiently hydrolyze GTP. It moves all the residues around, bumps out 5 water molecules, lines up glutamine 61, and then attacks the gamma phosphate. And that's what GAP does when it binds to the RAS protein. So you can easily see that if you mutate glutamine 61, this machine won't work. So those mutations in RAS are totally dead for GTP hydrolysis. Likewise, if you look at a slightly different view of the protein, this is the RAS stuff is in yellow here, and here's the arginine finger from GAP poking in here. Again, lining up all the residues of the active site perfectly for GTP hydrolysis. If you replace glycine with almost any other amino acid, it completely distorts the active site and prevents the arginine finger from lining up the catalytic residues. And that's the problem with the glycine mutations such as valine or cysteine or aspartate. These side groups prevent the arginine finger from correctly making the active site, and therefore it doesn't work as an off switch. Same is true of residues at glycine 13. That's why glycine 13 and 12 and glutamine 61 are the key residues involved in human cancer. They all break the off switch, so RAS now gets stuck in the active GTP-bound state. And that's the problem, which causes, by the way, probably 1 million cancer deaths a year are caused by KRAS mutations, if you include all the lung cancers worldwide. So that simple defect in the switch is responsible for a tremendous number of cancers. And again, as Susan said, in pancreas cancer and in lung cancer, RAS mutations are the initiating event. Pretty sure of that. Whereas in colorectal cancers, they are definitely not the primary initiating event, and that's true also in other cancers. So one of our efforts at the Frederick National Lab is to solve some of these key structures of mutant RAS proteins complex with their regulatory proteins or effectors. And surprisingly enough, when we started this project, we were somewhat, or many people were surprised to see that these structures never got solved. Back in the 80s, when people started working on the structure of RAS, they solved the structure of wild-type RAS and a couple of mutants, and then the field moved on. So we don't have any structures at all of any oncogenic RAS protein complexed with RAF, its major effector, or with GAP, its major regulator, negative regulator. So people just, the field moved on, people found other things to work on, but these structures were never solved. So we don't know yet whether it's possible to actually fix the broken switch because we've never actually seen it in a complex. So that's one of our major goals, is to actually solve the structures of mutant proteins bound to GAPs and RAFs and see if we can see there are potentially new pockets or new ways we might be able to find drugs to turn off the RAS protein. So that's a major effort. So again, no structures of KRAS have ever been solved. We also don't have any structures on full-length RAS protein with the lipid tail that Susan described. So tremendous amount of information that is missing if we want to really attack this as a proper drug discovery effort. In hindsight, everybody rushed into RAS in the 80s because it was the biggest oncogene and it seemed sort of simple. And then the farnesyltransferase drugs failed because of the backup system, the geranol transferase. And people moved on, basically, to target kinases and other cancer targets. But they never actually accumulated all the data you need to make this into a proper drug discovery program. And that's what we plan to do at Frederick. This is an updated version of the pie chart that Susan showed to show that in the mutations that cause lung cancer, the unknown pie is getting smaller as more mutations are found. And all the mutations that are found, really, in lung cancer map to the receptor tyrosine kinase RAS pathway. And these are all thought to be driver mutations. So except for the unknowns here, maybe 25% now, 75% of lung adenocarcinomas are driven by activation of the RTK RAS pathway, either by tyrosine kinases, RAS, or RAF, or things that affect the pathway less directly. And the newcomer on this pie here is the NF1 protein, the negative regulator of RAS, neurofibromin protein, which accounts for some 8% now of all lung adenocarcinomas. So this puts the NF1 protein up there as the third most frequent tumor suppressor in human cancer, behind P53 and P10. So loss of NF1 leads to hyperactivation of wild-type RAS, with the same clinical consequence as mutating the RAS by mutation, locks it in the active state. Just this little sidebar into some RASopathies. I find this fascinating, that the NF1 gene was cloned originally based on the fact that it is the gene responsible for the disease neurofibromatosis type 1. About half of these cases are transmitted through the germline, and about half are sporadic. Okay? But it's a pretty common disease affecting 1 in 3,500 people worldwide. And these individuals inherit or acquire a defective NF1 gene that can't turn RAS off. And the result of that is shown here. Many of these phenotypes, all these phenotypes, are due to hyperactivation of RAS through loss of NF1. This can lead to benign and malignant tumors, growth defects, and other disabilities. Sorry about the typo here. This gene was cloned in 1990 based on the family lineages in which this runs in the family. And it was found to be related to the class of enzymes that we discovered called GAPs that turn RAS off. And just to show the level of excitement when this was cloned and identified, this paper was submitted to Cell on the 24th of July, 1990, and it appeared in the August issue of Cell. So that's a fast turnaround. And it was sort of a very interesting paper because it meant all of these very heterogeneous phenotypes that have been well known to be associated with neurofibromatosis can all be put down, in theory, to just hyperactive RAS, because GAP was known to be a negative regulator of RAS. And then we followed up quickly, showing that this protein does, in fact, interact with RAS and negatively regulate it. So this led to tremendous optimism in the NF1 community that now this disease simply boils down to too much active RAS. We should be able to find ways of turning RAS down as a therapeutic strategy. But 25 years later, there's no approved therapy for any aspect of the NF1 disease. So we haven't been able to figure out how to turn RAS down and provide any clinical benefit for patients suffering from this disease. The NF1 protein is a gigantic protein, probably why it's so frequently mutated in human cancer, because any mutation which leads to a defective protein actually has a strong phenotype, hyperactivation of RAS. So the region of NF1 that interacts with RAS is this little yellow box here, the GAP domain. That's the structure I showed you earlier, bound to RAS with its arginine finger right there. The rest of it is unknown territory. There are no domains here except this membrane domain that show what it does. Even Saccharomyces cerevisiae has a paralog of NF1. Now, of course, yeast don't have tyrosine kinase signaling, so this must be a protein involved in something more fundamental than signal transduction from the receptor. So somehow I'm determined to figure out what this protein does normally, but I just haven't been able to figure out a strategy for doing that. Because when you mutate this protein, you just hyperactivate RAS and you get a very strong phenotype. But recently we got some clues as to how it gets regulated by looking at another disease which is often, or was actually associated with NF1, classified as an NF1 disease. So this is an NF1 disease in which the NF1 gene is wild type. So Eric Legius in Belgium decided there must be some other gene which phenocopies loss of NF1. And he actually found the gene responsible for these phenotypes. And it turned out to be mutations in a protein called SPRED1. So loss of SPRED1 phenocopies loss of NF1. So this was a great clue for us. We thought, okay, if we can figure out what SPRED1 does, that'll help us figure out what NF1 does. So we did mass spec on wild-type SPRED1 and some of the pathogenic mutants from this syndrome and found that, in fact, the major function we could find for SPRED1, it binds to NF1, and they actually work together as a complex. So these are now all the pathogenic mutations in SPRED1 from what is now called Legius syndrome, and they map to these different regions of the SPRED1 protein, and they all affect NF1 and its ability to interact with RAS. So basically, SPRED1 is a scaffold protein that takes the NF1 protein from the cytoplasm to RAS in the membrane in response to specific signaling. So without the SPRED1 protein, the NF1 protein just doesn't work. So now we have some new clues as to how to go after attacking this protein because we know a lot more about its biochemical function. And just another sort of sidebar, just reminded me that the NF1 disease, neurofibromatosis type 1, is a very common disease, but it's also part of a whole group of diseases we call rasopathies, because these are usually either inherited or sporadic mutations that lead to all kinds of developmental defects through hyperactivation of the RAS pathway in every case. So remarkably, there are kids out there who have Costello syndrome who have germline mutations in HRAS. And these mutations are full-blown oncogenic mutations in HRAS. So every cell in those kids' bodies has a full oncogenic HRAS mutation. And they develop learning defects and have all kinds of other problems during development. These are other versions of the same kind of problem. So hyperactivation of the RAS pathway to some degree is compatible with life, but it leads to a large number of developmental issues. Actually, one particular interesting one that we discovered was that CFC is the result of activation of BRAF, the melanoma gene. And some of these kids actually have germline mutations in BRAF that are the same mutations that actually cause malignant melanoma. But these kids don't get melanoma, and they survive, at least till early age. So it's very hard to understand how the same mutation and the same gene can be compatible with life on the one hand, and yet be a major driver of malignant melanoma in adults. Anyway, get back to the issue of the major mutations of KRAS that occur in these major diseases, as we've already heard. This is the number of patients that are affected by these alleles. So at the Frederick Group, we've focused on these proteins because they are the most frequent in human cancer. G13D is interesting because there is evidence that in colorectal cancer, patients with G13D colorectal cancer do actually respond to cetuximab, in contrast to the G12s. That hasn't been verified in a prospective study, but retrospective analysis suggests that there's some benefit in treating those patients. We have no idea why a G13 mutation should be responsive to EGF inhibition, but the G12s are definitely not. So there's biochemistry there we don't understand, but need to really understand who should be included in these treatments more efficiently. Also, these mutations aren't equivalent. In the old days, we thought these are just different ways of locking RAS into the GTP state because they all abolish gap-dependent hydrolysis of GTP, stick the protein in the GTP band state. But clinically, you can see that patients with mutations of G12V and C, they have a worse clinical outcome than G12D, which actually is the most common overall. And some speculation that G12D selectively activate these two RAS effectors, whereas C and V selectively activate this one. And again, 13D respond to cetuximab, G12s don't. So we have to start thinking now of each of these proteins as being somewhat different, not just in their severity, but also qualitatively in the way they actually turn on downstream signaling. Now, this doesn't make any sense to the old school structural biologists who looked at the structures of RAS and thought, well, the mutations occur on the sort of inner workings of the GTP switch. The outside surface of the protein is not going to be any different, whether you mutate glycine to alanine or valine or whatever. But these clinical data suggest that, in fact, there must be differences in the way these mutant proteins engage downstream proteins, such as RAF, PI3 kinase, and RAL-GDS. That's another reason we need to get these structures solved, because if they're different, there must be unique opportunities to intervene. So we can't explain these differences at all right now. This shows the structure of the RAS proteins. Again, the G domain, which I showed you, the beating heart, is this G domain, which is almost identical between each of the RAS proteins. And again, the differences, as we heard, are the C-terminal region of the protein, where these unique sequences are processed differently for each of the RAS isoforms. And this, you might think, is where the differences must be to account for the fact that KRAS has a different biology than HRAS and NRAS, for example. Because the effector binding regions of all these proteins are identical, and they all bind the same proteins, apparently with the same affinity. So biological differences must come from the back end, which is involved in processing. So we've undertaken an analysis of different RAS isoforms and different alleles of RAS using a sort of reductionist system of making cells which are identical in every way except the RAS protein that drives their growth. So we've taken the system developed by Mariano Barbacid in Madrid, where he was actually able to make mouse embryo fibroblasts RAS-less. And he did that by taking mice that have no HRAS and NRAS, because those genes are totally dispensable for mice. And he made the KRAS locus surrounded by sites that enable it to be flipped out using Cre recombinase. So you can make MEFs from these mice, and then you add tamoxifen to activate Cre recombinase, and you pop KRAS out. So now you have cells that have no RAS at all, no H, N, or KRAS. And they just stop growing and sit there, sitting on the plate for months until they either get infected or Stemness somehow. But they're remarkably stable. And we can rescue these cells by introducing HRAS, NRAS, KRAS in 2 different versions, or mutant versions of any one of these. So at the Frederick National Labs, in collaboration with my lab at UCSF, we've made a whole panel of these single isoform MEFs driven by only KRAS G12D or only KRAS 61Q and so on. And this enables us to really understand the differences between these proteins in detail, because there's no confusing elements of having multiple different RAS proteins. So this is just a cartoon to show how it all works. We've even been able to screen compounds in a sort of pilot study done by Matt Holderfield, Frederick National Lab, for compounds that selectively hit KRAS versus HRAS in an isogenic background. That really focuses on compounds that really get to the heart of the matter. And this just shows you one example of an ongoing study to show that these RAS proteins are different. So I was asked not to show too much primary data, but this is pretty cool. So here we've just taken these single isoform MEFs with these mutations, Q61L or R, or G12C, or G12D, and then simply immune precipitated these proteins and blotted for proteins that could bind to RAS. And the take-home from this is that if you compare this particular protein, the scaffold protein that holds RAF and MEK together, glutamine 61 mutations are very highly loaded up with this scaffold protein, but G12C proteins are not. So just at this simple biochemical level, you can see that these proteins engage different downstream complexes. And the RAF proteins from these cells are also very different. Again, if you look at KSR line, they bind to different proteins. So each of these mutants has different properties. That's really important if you think about trying to treat tumors in which you have KRAS mutations with, say, a drug that affects MEK, the MAP kinase pathway. Some of these mutants drive signaling through MEK, some less so. So that is a really important distinction which hasn't yet been made, that some of these may actually respond differently to inhibitors of downstream pathways. But in any case, we want to understand why these differences exist. So we talked a little bit, or Susan talked a little bit in the previous discussion about going downstream of RAS and blocking RAF and MEK. In the old days, when a lot of these drug discovery efforts started, say back in 1991, this is what the RAS pathway looked like. It seemed like a linear pathway, RAS, RAF, MEK, ERK. And that is probably true in primary MEFs. This is the major pathway that RAS turns on. But it would seem from this that in a cancer cell with mutant RAS, you've lost upstream signaling because RAS is stuck in the GTP state. You would have predicted that blocking RAF or MEK or ERK with small molecules, A, would have done the same thing since it's linear, and B, would have shut down RAS signaling, okay? And you'd have been wrong on both counts because these drugs don't work effectively on RAS tumors, as we just heard. In fact, if anything, they make it worse in some cases. RAF inhibitors actually activate the pathway. So the bottom line was that this pathway is sort of ludicrously simple. And it's based on genetic systems such as flies and worms and even mammalian cells that show the hierarchy of proteins correctly, but they don't show the details of how the whole thing is wired together. So for example, if you take 5 different RAF inhibitors with totally different structures and mechanisms of action and put these onto a KRAS-driven cell line and look for phospho-MEK, which is the readout of RAF kinase, you can see in a dose-dependent manner the RAF inhibitors activate RAF, exactly the opposite of what we expected. So in the absence of any drug, you don't see much phospho-MEK. And that's because this pathway is not on fire, as we expected. But when you add RAF inhibitors, the pathway comes to life. And so this is the drug that we developed. RAF is kinase is activated by the RAF inhibitor, as it is by each of these different RAF inhibitors. Nobody expected that. And it took several Cell papers, Nature papers, to understand what on earth was going on. But it just makes the point that this pathway, although it looks simple, is much more complicated than we expected. And just for those of you who care, this is what Matt Holderfield did at, who's now in the lab at Frederick Lab. He even showed that the purified kinase domain from wild-type BRAF, in this case, is activated in the test tube by RAF inhibitors. Okay? Just pure kinase is activated by the RAF inhibitor, and that's true of all these inhibitors. And the reason this happens is, for those of you who are biochemists, because RAF proteins get recruited to the membrane as dimers, as Debbie Morrison at NCI Frederick has shown. When RAS recruits this dimer to the plasma membrane, you get a nice signal of activation of MEK, although I should say we don't understand how RAS activates RAF when it gets to the plasma membrane. It certainly does, but the mechanism is not clear. But RAS recruits RAF to the membrane and activates it. They get a nice signal. But then, and especially with mutant RAS proteins, we sort of stay too long on the membrane because the off switch is broken. These RAF proteins start to talk to each other, and they turn each other off. So the RAF kinases autophosphorylate and negatively regulate themselves. Now you have basically inactive RAF. Until you come along with a RAF inhibitor, and the first thing you do is relieve this autophosphorylation, which is a negative regulator. So you relieve autoinhibition, and that leads to an increase in RAF kinase, when one of the RAFs is active and one is shut down. If you had enough drug, you can shut it down completely. So you get a bell-shaped curve. Okay? So this is why RAF inhibitors haven't worked for RAS cancers, because of this bizarre, paradoxical ability of these drugs to actually activate the kinase. On the flip side, though, RAF inhibitors have been effective, as I'm sure you all know, in malignant melanoma, where V600E RAF is mutated and drives the bus in a RAS-independent way. Totally rogue RAF kinase can drive these melanomas. We can treat these patients with RAF inhibitors to very high doses because in normal tissue, we don't turn RAF off. If anything, we activate it. So the therapeutic window is sort of infinite. So you can really clobber these tumors with RAF inhibitor without worrying about the effect on normal tissue because of its paradoxical activation, RAF inhibitors activate RAF kinase in normal tissue, not inhibit it. So that gives you a really good therapeutic window, which turned out to be essential for clinical benefits in malignant melanoma, because if you don't get 95% inhibition, it don't work. So you can really get a strong inhibition without therapy, without any collateral damage on normal tissue. That's a pretty unique situation. Not true of MEK inhibitors, which are next downstream in the pathway, RAF-MEK. MEK doesn't do this weird dimerization thing at all. So with MEK inhibitors, it just acts like a proper kinase. You inhibit it, the activity goes down instead of up. But unfortunately, when you inhibit MEK, as shown here, with a MEK inhibitor, you actually activate upstream signaling. So MEK inhibitors activate the PI3 kinase pathway through the EGF receptor. This is sensitive to gefitinib. And that's because RAS suppresses upstream signaling by feedback loops. As soon as you shut down the RAS pathway at any level, actually, you get hyperactivation of EGF receptor. And then cells can then activate downstream signaling and help them survive. So I know this is horribly complicated, but you tend to draw the MAP kinase pathway like this, simple linear pathway, but these feedback loops put the brakes on upstream. So persistent signaling from RAS leads to suppression of tyrosine kinase signaling. When you block this pathway with MEK inhibitors, you relieve negative feedback, and then EGF receptor goes nuts and activates the PI3 kinase pathway. And that offsets the effect of a drug. That's something else we didn't know about either. So you could say, well, if we give these patients enough MEK inhibitors, we'll shut down everything. But you can't, because MEK is essential in normal tissue. So for MEK inhibitors, there's a very narrow, if existent, therapeutic window. You can't shut down this pathway with MEK inhibitors, unlike RAF, where you had that unexpected ability to do so. Anyway, so going downstream has been much more complicated than we thought. And it's not over yet because people are still testing different combinations of drugs to try and prevent this rebound effect and so on. We're sort of chasing our tails a little bit because fundamentally we can't shut down this network effectively, completely, without being toxic in normal tissue. So it's sort of back to trying to understand the basics biology of RAS proteins themselves. That's the best place to intervene in the long run. I just to zip through one vignette. I know we have to stop at 5:30, but we've tested the hypothesis that KRAS actually is particularly frequent in human cancer relative to HRAS because it has additional properties that make it more malignant. And the properties that we've discovered are that KRAS makes cells more stem-like than HRAS. The KRAS turns on a whole stem cell pathway that makes cells drug-resistant, able to grow as single cells, and able to metastasize. HRAS does not do that. So it's a unique property of KRAS. And so we compared HRAS transformed cells, isogenic cells, H versus K. Everything's identical except the C-terminal regions of these proteins. We chose cells which have the same level of PI3 kinase and MAP kinase signaling and the same level of RAS GTP, and then asked, are these cells different in a more relevant biological context, such as growth in vivo or in 3D? And then we see the huge difference. Just focus on the bottom line here. KRAS is able to make single cells grow as spheres in a stem cell assay very efficiently. HRAS doesn't do that. HRAS is unable to form tumors efficiently when you implant a very small number of tumor cells into a mouse. KRAS is very good at it. So here's a mouse with KRAS on one side and HRAS on the other, and the KRAS tumors are obviously growing massively faster. KRAS also turns on drug resistance genes and stem cell markers. HRAS doesn't. So we think this is a completely new aspect of KRAS signaling that has therapeutic possibilities. So we looked at genes that are differentially regulated by KRAS versus HRAS. And the one that jumped out most obviously was a cytokine called LIF, which is a member of the IL-6 family. It's a cytokine that's commonly used to prevent embryonic stem cells from differentiating. It's used in, say, the Yamanaka cocktail for keeping mouse pluripotent stem cells from differentiating. Okay, I never heard of it when my postdoc discovered it. I went to Wikipedia and found this diagram that shows that LIF is necessary to keep pluripotent stem cells in this self-renewing state. If you withdraw LIF, you'd get differentiation. KRAS cancer cells pump out this cytokine that promotes stemness in the cells themselves and probably in the surrounding tissue, but certainly in the cells themselves. If we knock down LIF with shRNA in a tumor cell line, we significantly reduce initiation of pancreas tumors, and a monoclonal antibody that neutralizes LIF prevents establishment of pancreatic cancers. So we think LIF is an important element to establishing pancreatic cancers and other cancers, most likely, and then neutralizing LIF will reduce many aspects of pancreas tumors. Neutralizing LIF makes them less stem-like and therefore more susceptible to drug treatment. So neutralizing LIF makes pancreatic tumor cells sensitive to gemcitabine. I think it's shown here. Oh, maybe not. Yeah. Okay. Finally, another aspect of this, this stemness, we think derives from the ability of KRAS to bind to calmodulin, which it does so at the C-terminal region, which is the unique part of KRAS. So this binding actually results in inhibition of calmodulin-dependent kinase and suppression of the non-canonical Wnt signaling pathway. I'll just skip through that because I haven't got much time, but binding to calmodulin inhibits CAM kinase and prevents all kinds of downstream pathways and affects pathways that affect stemness. So the point of all this is that if we can prevent calmodulin binding to KRAS, then we can reverse the stemness features of human tumor cells. This interaction is regulated by phosphorylation on serine 181, which is a protein kinase C site. So as Peter Blumberg, NCI, has shown, there are a number of ways of activating phosphorylation of this serine to prevent calmodulin binding. We can make a mutation of the serine to make it look like a phosphate. It doesn't bind calmodulin anymore. That mutation in KRAS prevents the stemness properties that I just described and reverses malignancy. So we can turn KRAS off, not from the proliferative point of view, but from the stemness point of view, okay, by inhibiting by preventing calmodulin binding to KRAS. And skip a couple of slides at the end. The therapeutic value of this that we're now testing is that we've actually got hold of a compound called prostratin, which was identified as an antiviral agent by sort of analogy to your last slide. An antiviral agent that was identified by the people of Samoa as an active ingredient in the bark of the marmala tree, which the people in Samoa use as an antiviral. And a group of investigators from the NCI discovered that it has an active ingredient, which is a PKC agonist called prostratin. And it's actually heading towards clinical trials as an antiviral through this discovery. But prostratin activates phosphorylation of KRAS on this C-terminal amino acid. Prostratin treatment of mice prevents pancreatic cancer and actually reverses the phenotype of established pancreatic tumor cells. So here we have an antiviral drug which actually may be very useful in treating pancreatic cancer. So this is something which we're currently testing in the lab. But sort of the bottom line is that although we thought we knew a lot about RAS and we thought we knew everything about RAS, there's a lot of details we didn't understand at the protein level, the signaling level, and even the biological level. And we now think that these new insights can be translated into new ways of treating KRAS cancers. Thank you very much for your attention. Thank you. Thank you.
So what would be your preferred method then to target RAS? You alluded to half a dozen. So what do you think is most feasible and most likely to succeed and have broader applicability?
Well, as of today, I would say based on what we know today, I think the results that we've obtained with prostratin look like the most best short-term bet. This is an orally available drug, a compound, which is safe and does exactly what we hoped it would do. It prevents metastatic features of KRAS cancers. So that is something which is brand new, not even published yet, but it seems to me that kind of approach will definitely go to the clinic with it in the next few years. It should, I think, have some real benefit. Neutralizing LIF also looks pretty good to me. Longer term, I think understanding the structures of the RAS proteins and their complexes has the best opportunity presents the best opportunity for finding small molecules that will kill RAS by preventing RAS signaling. Then there are also the approaches of targeting the KRAS gene itself using siRNA that can be delivered systemically. That's another potential opportunity. Or enhancing the immune response to KRAS tumor cells, which is another option. So I think we're looking at probably, as you say, 7 or 8 different options, all pretty early for the most part anyway. But we're betting on 4 or 5 of them at the Frederick National Lab, and we'll assess the situation in a couple of years and see which horse is looking most promising. And also hope that other people in the biotech and pharma world will work with us on picking up some of these different approaches and taking them across the finishing line.
Go ahead.
Hi, I saw that you showed data to suggest that the different mutations in RAS will select for certain downstream effectors. Do you think that this might also be occurring in tumors that are termed like oncogene addicted, that are addicted to the KRAS oncogene, as opposed to tumors that happen to have the mutation but are no longer reliant on it for their tumorigenesis?
Yes. Well, you're raising an interesting question. Which tumors are actually addicted to KRAS? So far, in pancreas cancers at least, it seems like they're totally addicted to KRAS in the experiments that have been done where it's been taken out. So I would argue that at least for KRAS and lung cancer, they remain addicted to KRAS, really. Cell lines, maybe not. In fact, there are cell lines, I'm sure, that even pancreas cell lines that don't care about KRAS in cell culture, but you put them into 3D or into mice, they don't grow at all. So they are addicted. But it's a very interesting question as to whether or not addiction will be different for the different alleles, and that hasn't been done. Those addiction experiments have only been done on 1 or 2 alleles, so it don't know yet. It's a great question.
I was wondering if the cross-phosphorylation of RAF could be targeted, because for achieving cross-phosphorylation, they should be at some point in a physical contact or a dimer formation. Is there any evidence for that, or is there any effort to develop some sort of therapeutic approaches?
Yes, indeed. As I mentioned, Debbie Morrison at NCI Frederick is actually planning on doing screens for compounds that prevent RAF dimerization, because dimerization is essential for activation. So those screens are not too difficult to do technically, or so it seems. But that's a very interesting approach. And she's shown by mutagenesis and proof of concept experiments that preventing RAF dimerization prevents downstream signaling. So those efforts are underway.
I wonder whether in regenerating tissue or rapidly turning over tissue, the bone marrow, the liver after partial removal, is there a transient phase where any RAS, is expressed at a time particularly early on, when in like the regenerating liver or the primordial cells in the bone marrow, when they show drug resistance and stemness-like characteristics? Is there a relationship to RAS presence or function?
Well, it'd be easy to say yes, because I'm sure that's true. But I don't know of any evidence to support it, because RAS proteins are expressed everywhere in all tissues. So the question would be, are the different isoforms active during those states, and whether that activity translates into effects on stemness. I just can't tell you at this moment.
But I mean, are they overexpressed, underexpressed in relationship to this kind of rapid proliferation?
I don't think so, no. I think in most tissues, RAS proteins are kept in the off-state GDP bound. So, they have a tremendous capacity to be activated by upstream signaling and by inhibition of gaps and so on. So the RAS proteins are active by nucleotide binding and not by increased expression for the most part. That's much more difficult to measure than just total levels of protein. So I think it's just a harder question to answer, because it's difficult to measure RAS activity, really, in that kind of way. You can do knockouts in mice, as people are now doing, and then show that different tissues depend on different RASs at different stages by genetic ablation. I think that is one way to get at your question.
I think
great question. NF1, certainly. About 10% or so of patients with NF1 succumb to malignant tumors. It's not 100% by any means. The Costello kids have a higher frequency of rhabdomyosarcomas and one other tumor type. I've forgotten what it is, actually. But the BRAF mutant kids don't, which is totally bizarre to me. Noonan syndrome kids, Noonan syndrome is caused by activating mutations of SHIP2 or weak alleles of KRAS or activation of SOS. Those kids have a high frequency of juvenile myelomonocytic leukemia. So I think generally, yes, with some sort of odd exceptions.
Or maybe Susan. So in the absence, temporarily, of something that would be able to block RAS in RAS-expressing cancers, so what is the rationale to combine different drugs that will be acting downstream from RAS and preventing perhaps the EGFR activation, say, F3 kinase inhibitors together with mTOR and MEK inhibitors? Is it rational? Do you have any evidence of activity?
Well, it all makes sense from the point of view of the RAS cancer, but the problem is that where's the therapeutic window? I think that is the problem. I think a large number of trials have been tested with EGFR inhibitors plus MEK inhibitors, or AKT plus MEK inhibitors, and every combination thereof. And as we heard from Susan, there are some signs of some activity, but you just can't dose enough to shut down that whole network without affecting normal tissue. You can in mice, enough to sort of encourage you to go forward. But I think toxicity of all those combinations has been a real problem. So there are people like Neil Rosen and others who at least have some theories as to how to get around that kind of issue by different dosing schedules which prevent the rebound effect and get the maximum sort of bang for the buck before these rebound effects chip in. And that can maybe will improve things, but I don't think personally that those combinations are going to get us where we want to be, because we're working against a therapeutic sort of gradient. RAS tumors downregulate, everything that we want to target.
So in related to that, so the old days, there was some work Mariano Barbacid and many years ago, Josef Schlesinger, showing that some of the RAS mutations require EGFR.
Yeah.
How do you relate to that? Is it?
Yeah, that's a very good point. I didn't have time to go into this in any detail, but turns out that the G12 mutations of KRAS actually do have significant intrinsic GTPase activity. So in these RAS-less MEFs that I showed you, we can show that they actually respond to EGF, and they respond by GTP loading from a relatively high baseline of maybe 20% to 100%. It's a pretty big increase. So they actually depend on upstream signaling. They're not totally independent. Whereas the glutamine 61s, which have zero GTP activity, are always GTP bound and don't care about EGF at all. But the mice that Mariano used for those experiments were G12V, which does have a big window of activation. So my feeling is that they're only, on their own, they're only partially active. They need help from upstream signaling.
So you can co-target upstream?
You might think so, but they depend on EGF less than normal cells do. So they still depend on it, but it's the therapeutic window is, again, going in the wrong direction. So, there may be a way there somewhere, but it'd have to be more specific than just that. Great question.
Wait a minute. Here,
Hi, thanks, Frank. A strategy I think several groups have looked at is using synthetic lethal screens to try and identify pathways or proteins you can inhibit that would only kill cells that have a mutant RAS. Mm-hmm. What is your feeling on that as an approach? I guess they had some initial promise, but kind of faded away. Are they being revisited? Are there solutions to this?
Well, we are revisiting it. We put out an RFA, actually, based on discussions we had at the the Frederick National Lab, to get people to come back again and have another shot. Because, as you point out, the first round of synthetic lethal screens really didn't work. So all kinds of issues. Partly siRNA or shRNA wasn't clean enough, with too many off-target effects. Cell types were sometimes, the results were sort of idiosyncratic and couldn't be reproduced and so on. So there's a whole number of people have reapplied to try and take that to the next level, where the reagents are much cleaner, and we learned a lot from the first wave. Now let's try again with the second wave, using either better libraries, CRISPR instead of shRNA, or targeting cells in 3D instead of 2D, and so on. So the next 2.0 versions will soon be tested, hopefully. Because the idea of finding, in an unbiased way, genes which are essential for KRAS survival without being biased by these network PowerPoints that we draw, I think is a great idea. We just haven't found any yet.
Okay, well, listen, I want to thank you both for a very exciting presentation. It was very, very exciting. Thank you.